Here's a tutorial on how the loop of Henle achieves osmoregulation. So just a quick recap of where we are. We started with ultrafiltration, which was taking place in the cortex of the kidney. And in that process we had filtered through the glomerulus. And we'd got a lot of things going through into that filtrate. And then the filtrate moved still in the cortex. So that was ultrafiltration in the glomerulus, which is part of Bowman's capsule. And then further along, we went into the proximal convoluted tubule, the PCT, where we had selective reabsorption. So that was taking back things like glucose, salts, water, amino acids, and small proteins. So the next part is we're now going to dive down into the medulla of the kidney. And the medulla of the kidney is where we're going to have water absorption. So this part of the kidney, the medulla, it's the very salty part. So the other labels, that was the pelvis. So we haven't yet made urine because this is where urine's going to be collected. So this part here, the medulla, is very salty. It's where we're going to get water absorption again. So we're going to get more water absorption. We already had some water absorption in the proximal convoluted tubule, and now we're going to get more water absorption in the medulla, in the loop of Henle. And then there's going to be final water absorption still in the medulla in the collecting duct, but we'll see that afterwards. So here's a little flow through. We've been in Bowman's capsule for ultrafiltration, proximal convoluted tubule for selective reabsorption, and now into the loop of Henle for osmoregulation. Where that is going to, it's about balancing the water content. In the distal convoluted tubule, we get some further selective reabsorption. We also have secretion of toxins. And overall, it can be said that it adjusts the pH. And it also adjusts sodium, potassium levels. So one of the keys that we need to remember here as we're going through is what salt and sugar do, and this process down here. So stop the video and see if you can label all of the features down on this diagram and predict which way water will be moving and why it will be moving that way. Down here we have two semi or partially or selectively permeable membranes. So this is an example actually given with visking tubing, which is dialysis tubing, which mimics the kidney. So in these two situations here, we have solute, which could be salt or sugar. And we can see that we've got a higher solute concentration here. And a lower solute concentration over there. So what we're not seeing on this diagram is that when a solute dissolves, it means that molecules of water interact with it due to polar attractions, remembering that water is a polar molecule. So it must mean that these solutes have charged regions. They have charges. So things like glucose and sugars are going to have charged regions. Now that means when water is in an environment with solute and salts and sugars, the water will attach, and if the solute cannot move through the membrane, if it's stuck in place, that means the water's stuck in place. So all the other water is going to continue to move randomly and spread out randomly. But over here on this side, we're going to get water being stuck around the outside of these solute molecules. And that effectively reduces the concentration of free water molecules. And that means we'll get water moving overall as it gets stuck to the solute. We will have more water in this region over here, and that leads to the rise in the water level. So this is a key principle. Where we have salt, water will move. It will remain because it's stuck to the solute, and then it's unable to freely pass back through. The solute can't pass back through freely, and so the level on that side rises. So what does this have to do with the kidney? Well, in the kidney, as we go down into the medulla, it gets saltier. And it's salty because of sodium, predominantly sodium. And it's going to get salty, very salty, because we're going to have some pumping of salt down here. So the kidney is pumping salt into the deeper regions of it. So it's going to get increasingly salty as we go down. Where we get salt, water will follow. So water will move to follow the salt. And that means that we can absorb the water. So this is our view of the whole structure. And let's remember, keep reminding ourselves that this whole structure is the nephron. So whenever we're talking about adaptations, functions, features, things of the nephron, we're talking about all these parts. We're talking about, we've seen, so let's try and label all of these. You might want to stop and see if you can label everything here. So here's the diagram completely labelled. We see the glomerulus, the knot of blood vessels where ultrafiltration takes place, surrounded by Bowman's capsule, which collects the filtrate, leading down into the proximal convoluted tubule, down to the loop of Henle, and back up to the distal convoluted tubule, and then to the collecting duct. And the whole structure of the nephron is surrounded by the vasa recta, which are blood vessels to collect the absorbed material. Okay, so arriving into the loop of Henle is the filtrate. The filtrate is still containing quite a bit of water, and it has salts, it could have toxins in, it is going to have urea in, and it should have very, very small levels of glucose, almost none at all. So it arrives there, and now the kidney wants to reclaim back some water. So we talked about it getting saltier as we go down. And that means that water will move out via osmosis. And it's going to move into the tissue fluid which is surrounding. And on both sides there are going to be blood vessels. So it's going to move into the blood and be taken away. Now you can see that the water is moving, the filtrate, is moving in one direction, and the blood is moving in the other direction. So this is called a counter current. And this is great for maintaining gradients, because as soon as some water has moved into the blood, it then moves along and is replaced with fresh blood. And this is the same as well if we're thinking about sodium being reabsorbed. As soon as sodium moves across into the blood, and perhaps the concentration of that part of the blood would go up, then it moves along, and now we get more fresh blood coming along. So the countercurrent maintains the concentration gradient. Okay, so it maintains concentration gradient. So what I've drawn here is a diagram representing the concentration of salt, and in particular the concentration of sodium. So we see that as we go deeper into the medulla, down to the base of the loop of Henle, we get increasing concentrations of salt. So this means as we move down, more and more water can and will move out. To understand why that's important, we need to think about what's in the filtrate. So the filtrate has water in it. There wasn't all the water reabsorbed. But as we go down, more and more water will be, so if we take that as being a kind of an area, a box, with a concentration, down at the bottom, we will have absorbed a lot of the water out. Now the other thing which would also be in the fluid, the filtrate that's going into the loop of Henle, is there will still be some salts. So I'm going to draw 1, 2, 3, 4, 5, there's 6 there. Now those salts could potentially be being moved as well. And so here's what the loop of Henle does. It puts a barrier to the salt. The salt, so this is a barrier to the salt. The salt cannot move. And now if the salt cannot move because of this salt barrier, the salt's stuck there. So I'm going to do 1, 2, 3, 4, 5, 6. The salt is stuck. It can't move, but we said that the water can move out. So water's moving out. And so we can see why this is important to have a very salty kidney, and also why we don't want to move salt out in the limb that's going down. This is the descending limb, because it's going down. So in the descending limb, water moves out because of the salt concentration. As we get down to the bottom of the kidney, it gets saltier and saltier. Now that's a real advantage down here, because even when we've absorbed lots of the water out down in this box here, meaning that we have an increased overall solute concentration because we've removed the water, we will still be able to absorb some of the remaining water. So the whole point of the loop of Henle is to maintain a very salty solution in the kidney. And that means to maintain a hypertonic. There is always going to be more salt here than there is in the filtrate. So it's hypertonic so that it could absorb the water. So what's going to happen by the time it reaches here? It's reached the very bottom and now it's starting to go to the ascending limb. So it's starting to go up the ascending limb. What's going to be here? Well, we're going to have almost, and I'm just going to draw one, almost no water. We've absorbed so much water that really all that's left in that, it's a very salty solution. And so we're going to switch round the case in the ascending limb. So as this fluid turns the corner here, now we are going to pump salt. So the salt from this very salty solution is going to be pumped out. And this is actually what generates the hypertonic nature of the medulla. By pumping, using sodium pumps, we can now build up a very salty concentration. Now, in order to rebalance the fluid, though, we don't let the water move. So I'm going to draw myself a barrier to water. Water is stuck in there, and sodium can move. Now let's think about what's going to happen then. So, well, firstly, like we said, we're going to get a very salty bottom of the loop of Henle. And then we're going to get more and more of the salt pumped out. And so if we think about what's going to come out the top, well, if we've pumped out a lot of the salt, we will have much less salt remaining. And we kept water, any remaining water, which we said was very little. Maybe let's draw 2. Then what we've done is we've restored the concentration of that solution. And so it's actually equal in the proximal convoluted tubule, where it started to then enter the loop of Henle, and the distal, which is what comes after. Now, I don't think if we count these two boxes here, box 1 entering and 2, that it's exactly the same. Here it's about 1 to 1, 1 solute to 1 water, and it's not quite the same there, so I haven't done it perfectly on my diagram, but they are equal by the end. So why is this important? Well, it means that once this fluid leaves, we won't have a very salty solution, which would then reabsorb water from the tissues around into there, and it would actually dehydrate the kidney. And it means as well that we've absorbed lots of water in the process. So we've absorbed lots of water, we have pumped sodium as the mechanism for making the kidney hypertonic, and we've then restored the solute concentration of the filtrate, which is going to enter the distal convoluted tubule. Okay, so here's a quick recap of what I drew before. So we have entering in, let's do 1, 2, 3, 4, 5 water, and let's do 5 salt. Now in reality they wouldn't be even, but as the filtrate enters, some of the water will now start to move out and be absorbed. But this region here was impermeable. There was a barrier. The descending was impermeable to salt. So the salt couldn't move, but the water could. So let's take some water out, but all of our salt remains. So all the way down, we've got more and more absorption of water, giving us a very salty solution. Whereas when we turn the corner, so let's remove even more, we've got our 5 salt, but then we get pumping of the salt out and into the medulla. And then by the top, if we think about relative concentrations, we would be back to having the same relative concentration that went in. So the ascending limb was impermeable to water, so the water stuck there. But by pumping out salt all the way through active transport, we now have restored the concentration in the PCT, the proximal convoluted tubule, and the DCT, the distal convoluted tubule, are going to be the same. So here's a picture showing it, and the technical way to talk about concentrations, solute concentration, osmolarity, is milliosmoles. So you can see here, as we go down, we get up to 1300 milliosmoles at the bottom, very salty. So we get the loss of water all the way down, but salt is stuck, so the descending limb loses water or absorbs water. Whereas in the ascending limb, water was stuck and we got salt being pumped. So it loses salt, and in the end, these two, the proximal convoluted tubule and the distal convoluted tubule, will equal each other. So the final thing to talk about what's happening in the distal was that we could get toxins being added in, and it also does some further balancing of pH because of it can absorb H+ ions, and further balancing of Na+ and K+ ions. So final review with the flashcards. We said that the filtrate entered the descending limb of the loop of Henle, which was only permeable to water but impermeable to salt, and therefore water would be drawn out by osmosis. In the ascending limb, there were pumps through active transport to pump sodium into the tissues of the medulla, but it was impermeable to water. And that, in the end, leads to a decrease in the filtrate concentration. And when we're talking about concentration, we're talking about decreasing the overall concentration of solute, because it was very salty at the bottom of the loop of Henle. And now, by pumping out sodium, we decrease the filtrate or the solute concentration, returning it to the same as the proximal convoluted tubule. And we also said, due to this pumping, we get more sodium at the base of the loop, deeper in the medulla, and surrounding it would be capillaries, the vasa recta, to reabsorb, to reclaim that water. Finally, in the distal convoluted tubule, we had readjustment of pH and of sodium, potassium, and hydrogen ions.